what is mRNA? And just to frame this up, we're speaking to a lay audience. Even though you're a card-carrying molecular biologist, and I once was, maybe I still have a emeritus license as a molecular biologist. I don't know. But we have to simplify this, not dumb it down, but simplify it. So the metaphor that there's a kind of, how do you express these things to somebody that hasn't taken cell biology or biochemistry or even organic chemistry? And the way that I deal with it is I reach for two different metaphors that everybody understands. One is a fried egg, and the other being a metaphor for the cell. And the other is the knowledge that we all have right now about digital information systems, what we call computers. And so my wife doesn't like these metaphors, but it's the best I've come up with. So if we think about how digital information gets processed, you have to have a place that you store the information. And then you have to have a way to transmit that information down to technologies that operationalize it. You could think of it as robots. So one way to think about this whole space is you have sort of a hard drive that stores information and repeatedly makes it available for reading and processing. You have a technology that's necessary to transfer that information out into the area that it needs to be used. And then you have a series of technologies. These are all biologics that have evolved over time, or depending on how you want to look at it, the creator generated. But in any case, they exist, however they came to be. You have a kind of a biologic technology in the cell that takes that information and turns it into operational output that we call proteins. And RNA plays a central role in all of this. Of course, deoxyribonucleic acid, DNA, is a double-stranded storage form of information that, unlike in the digital world where we have zeros and ones, it's a binary system. In the case of molecular biology and biology, all biology that we know, organic biology, you basically have a system of 4 different information bits akin to zeros and ones, which in the case of DNA abbreviated as A, T, G, and C. And you need to have a way to ensure that this information maintains integrity through time and across generations. If you want to take Richard Dawkins' point of view, that we are all basically vehicles for transmitting DNA through time, you want to make sure that that DNA maintains integrity. So what are you going to do? Well, it's what has evolved or been created is a amazingly a robust system involving 2 copies of the DNA that wrap around each other in the notorious double helix of Rosalind Franklin, Watson, and Crick. And so that double-stranded DNA is akin to the storage on your hard drive. It maintains it not only within the cell in a way that has mostly integrity. We can go into the nuances of that, having to do with various forms of DNA parasites and recombination and other things. But basically, you have this storage form, and then somehow that storage form needs to get copied, that information, and moved out into the area where it actually does its business, gets turned into something functional. So now the fried egg metaphor. You can think of, everybody knows what a fried egg looks like. And so the yellow part of the fried egg, you can think of that as akin to the nucleus of a cell, which is where the DNA resides. And just if you imagine a fried egg, there is a membrane, a barrier that defines the yellow part from the egg white. That, in the case of the cell, is the nuclear membrane. And the nuclear membrane has specific holes or pores, and it actively facilitates and controls what information can get in and out, what molecules can get in and out of that protected space where the DNA resides. And one of those molecules is RNA. And it's not just any RNA. There are multiple different forms of RNA that are copied off of a DNA template. And these include various structural RNAs, as well as a message ribonucleic acid. So mRNA is messenger ribonucleic acid. It's a single-stranded copy that gets complicated. It's processed. It's not necessarily collinear with the DNA, but it starts out collinear as a copy of the DNA, and then it gets processed in various ways and exported. And it goes out into the cytoplasm in the fried egg, that's the egg white. And there it encounters literally a little nano machine, a nano robot that takes that RNA like a ticker tape, and it runs that RNA through a kind of a pore or a groove in something called a ribosome, which is the little nano robot that makes protein based on the RNA code. And so what about the building blocks of the protein? So that just to go back just a step, in that metaphor of the little nano robot that takes the RNA, this is not meant to be sexual, takes the RNA that's feeding through that groove and is being used to build the protein, that the scaffold for that very structure is another kind of RNA, okay, ribosomal RNA. So we have messenger RNA, we have ribosomal RNA, and then we've got, well, how are you going to get the subunits of the protein, these amino acids? How are you going to kind of connect those amino acids in a structured way into this growing molecule that's based on a RNA template? Use the same basic biochemistry of pair bonding that is involved in the creation of the double helix and the transcription or making of the RNA, which is basically a copy. Use that same basic biochemical technology to couple. So you have an amino acid that is tagged onto another kind of RNA, transfer RNA, that has a 3-base connector socket you could think of. And so as you have this ribosome, that's this thing, composed of ribosomal RNA, it's the little nanorobot, and you have this message of RNA that feeds through it, that message is read 3 bases at a time, 3 of these letters at a time, by a transfer RNA that comes in, is attached to a amino acid, reads those 3 bases, says, oh, this is where I'm supposed to be. And then there's a biochemical connection reaction. And that gives rise to proteins, which is kind of the functional molecule that builds everything else in the cell, from lipids through enzymatic reactions, lipids, carbohydrates, all the other kind of stuff that has to go on in a cell, all gets built. And by the way, so does the DNA. It comes back. The DNA gets replicated using proteins also. So that's my metaphor. I hope that made sense to your audience. And it's this whole mRNA is thrown about as an acronym that confuses people. It's really simple. And the M stands for messenger. R stands for ribonucleic acid, which is just a string of biochemical linked subunits that form a linear polymer. But in RNA, it's a little weird. Here's a nuance. RNA folds up on itself. And so you can't, it's nice, you can look at the pictures in the textbook that an mRNA is just a string. That is a gross simplification. It turns out and this is kind of the essence of the RNA world, is that this molecule is super complex. It is wicked hard to predict what its actual structure is, because this folding and assembly is very much a function of the environmental conditions under which it exists. And it's very dynamic, and it's influenced by things, this is a read ahead, like the presence of pseudouridine molecules, which are modified bases within the RNA that are produced by a protein enzyme acting on the RNA that affect how it folds, how it functions, how it interacts with the ribosome, and all this other stuff. And of course, pseudouridine is a key property that was introduced by Karikal and Weissman on top of the technology that I was involved in creating back in the late 80s.
Now I want to, I would add one thing: the semiotic nature of what you were describing there. Essentially, you're describing what we call translation. And a good thing, I always impart this on my students, is that in biology, science in general, but in biology, there's a certain language that we use, and there's a reason why we use those languages. And no pun intended, we're using the terms translation because you're translating from one language to the other, which is from the language of DNA into the language of amino acids and proteins. And no matter how many times I hear somebody describe this process, I'm just blown away at its beauty, its simplicity, and its complexity, and its importance in almost everything to do with molecular biology. So, and I'm also very happy that you mentioned Rosalind Franklin, because she doesn't get her dues all that often.
Yeah, there's like 5 or I think maybe even 6 or more Nobel laureates involved in getting us to this very simple story that we just covered in the last 5 minutes.
And in my opinion, you should be one of them too, but we'll get to that later.